Hi everyone, this is Himanshu, and in this lecture we will start with the gene therapy. The gene therapy involves the removal of the defective stem cell from the patient's bone marrow. So let me write it here that it means that the removal of defective stem cell from patient's bone marrow. So basically the stem cells are removed, are genetically modified for the correction. So that's generally what happened. And the stem cells, then we will discuss them. Okay, fine, we'll discuss them right on. So what generally the stem cells are, let us write it here. So these are the most basic unit of an organism in a cell. So as we know that cell is also the most basic unit of an organism, and it is the smallest unit of life. And the cell are actually of the two types. So if I write it here that the cells are of the two types. The first one is what we call as a differentiated cell, and the other one is called basically the undifferentiated. Differentiated cells. So these are the two classifications of the cell. The differentiated cells are those building blocks of the tissues and organs, and they are specialized to form the tissues and organs. So simple as that, because we know that cells, when they combine together, they form tissues, right? So the differentiated cells are those cells which are specialized to form tissue or organ. Similarly, the undifferentiated cells are those that have the potential to develop into many different types of cells, like those in the muscles, the kidney, liver, etc. So those are the undifferentiated cells. They have the potential, to develop into many different types of cells. So this is a basic difference between differentiated and the undifferentiated. Now, when we say a stem cell is actually, they are the undifferentiated cells, So yes, so what we can easily say in layman terms that stem cells are the undifferentiated cells. Means they can be found in muscles, they can be found in the kidney, they can be found in the liver, hearts, lungs, brains, genitals, anywhere. So these are what the undifferentiated cells are, and this is what the stem cells are. Now these stem cells are also the two types actually. The one of them is what we call as the embryonic stem cell, and the other one what we call as a somatic cells. So that's how it is different. Now the embryonic stem cells, these are derived from the embryos. So these are derived from embryos, and more importantly, these are totipotent. Now the totipotent are a classifications of those cells which can produce a clone of the entire organism. So this is what we call as a totipotent. So I repeat, the totipotent is a term which is given to those which can produce a clone of the entire organism. This is what we call as a totipotent. Similarly, the somatic stem cells, or we can also call them sometimes the adult stem cells, these are the undifferentiated cells which are present in the differentiated cells in a tissue or organs. So I repeat again, these are undifferentiated cells, I'm writing the UC as that, which are present in differentiated cells, I'm writing it as DC, in a the tissue or organ. And most importantly, they help in repair and maintenance. They help in repair and maintenance of specific tissue or organ where they are present. So this is the difference between the embryonic stem cells and the somatic stem cells. So this is what it is. There's the one more category, what we call as an iPS cell. These are basically the induced pluripotent stem cells. So you can, because it was in the news, so it would be better to discuss this thing at least in a bit. So something called the iPS cells. These iPS are basically induced pluripotent cells. So this is what they are, and they are basically the adult cells. Adult cell means the somatic cell that we talked about here. So these are basically the adult stem cells in the umbilical cord or the bone marrow cells that can induce to show some properties of the stem cells. Means originally they are not, but they does show some properties of the stem cells. So they are generally used in the therapeutic cloning. So let me write it here. They are generally used in the therapeutic cloning to treat the degenerative diseases, degenerative diseases like diabetes, Parkinson, Alzheimer's, etc. And these are actually found in the bone marrow itself. And yes, this is basically mainly about these things, and they are created by actually the stimulating the mature and already the specialized cells back into the juvenile state without the need for an embryos. So this is mainly about the stem cells. Now the other thing that about the stem cells are that how they are actually significance, or you can say, as I told you, the significance of the stem cells, that they are basically currently available for the Parkinson disease thread and degenerative diseases. They are very much good in curing these things. And this process is what we call as a stem cell therapies. And more importantly, the potential use of stem cell can be the removing the baldness, the blindness, deafness, the amyotrophic lateral sclerosis, which basically occurs in the arteries regarding to heart, the myocardial infarction, muscular diabetes, even if you have a missing teeth, we can do some mutations with the stem cells and remove them and add them, then we can easily have your missing teeth growing again. With the bone marrow transplantation, spinal cord injury, etc., etc. These are multiple of them. So even if they ask in the prelims, then you can mark all of the above, because generally this is what happens. And this is what it is. Now next thing that we need to talk about is about the mitochondrial DNA and the nuclear DNA. Now see what generally happens. The DNA we have till now present in the body is in the nucleus, right? Now till now whatever we have discussed so far is about the mitochondrial DNA. So the some DNA, let me write it here, some DNA is also present mitochondria. Now actually during fertilization, The nuclear DNA, which is actually the 46 chromosome that is formed when the 23 chromosome is inherited from the mother and 23 from the dad. The mitochondrial DNA is the one which is, which comes from the mother. So it comes from mother. And this mitochondrial DNA is actually responsible, for the specific protein's metabolism. So we can say this chromosome, mitochondrial DNA, is actually responsible for the protein's metabolism, whereas the one which is received from the father is actually responsible for the inheritance itself. So it's a mother mitochondrial DNA, and it is responsible for the protein metabolism, and it is present in the mitochondria. It is different from the nuclear DNA because that comes from the dad and means father, and it basically responsible for the inheritance. So this is basically a difference between the mitochondrial DNA and the nuclear DNA. Apart from that, there was also in the news regarding the 3-parent baby technique. 3-parent baby technique. Now, sometimes what happens that as we know that the chromosomes pair comes from both the mother and father, and sometimes what happens, there is the mitochondrial DNA that is from the woman, it is sometimes defected. Maybe that woman is suffering from a disease. So because of that, the chromosomes, the pairing doesn't happen very much good, and the fetus which is born, it has some kind of defects. So in order to rectify such thing, what generally happen, now we have a man, we have a woman from whom we have a chromosome, but what generally happen, there is a donor woman also here. Now donor woman will provide a mitochondrial DNA, and that mitochondrial DNA will also be transferred to this thing. Now because of this technique, the fetus which was been born, now it will be the free from any problems and the defects. So this is what the whole procedure is called, 3-parent baby technique. So this technique involves removing the faulty mitochondrial DNA which was there in the actual mother and the nucleus from the mother's egg, and the resultant egg fertilizes with the sperm cells of the father outside the body in vitro, means outside the body it will be developed. So this whole procedure is actually what we call as a 3-parent baby technique. So in the next lecture we will study more about the gene editing and the gene drive technology. So let's jump to the next chapter. Thank you guys.